Now that we know the parts of the neuron, we're going to start to examine how it works. And we're going to begin by looking at what's called the membrane potential. And basically, this is just the difference in the electrical charge between the inside and the outside of the membrane surrounding that cell body. And that's going to be determined by all these ions flowing in and out through different types of channels. So we can actually use a machine called an oscilloscope to record the difference between the inside of the neuron and the outside of the neuron by putting these little micro electrodes in and outside in that extracellular fluid. And it's going to record the voltages of each of those and determine the difference between them. We call this the membrane potential. And when the neuron here is at rest, when it's not firing its action potential down the axon, we call that the resting membrane potential. And it's established by two different types of gradients. We're going to be looking at chemical gradients, and we're going to be looking at electrical gradients. There's a difference in charge, which we just said, between the inside and the outside, and that's determined by three different things. One is going to be the permeability of the membrane, how easy it is for those ions to be going in and out of that membrane. The second is going to be the action of the sodium-potassium pumps. These sodium-potassium pumps are going to be in just about every cell in your body, and they're going to be putting 3 sodiums out of the cell for every 2 potassiums it comes in. So it's constantly making the cell a little bit more negative and managing those sodium and potassium ions. And then it's also going to be determined in large part by the negativity of charged molecules. So we see that this is about negative 70 millivolts for its resting membrane potential. So that means there's got to be a lot of negative ions in there. And certainly potassium is not one of those ions, and sodium is not one of those ions. So we're going to see what causes that in a few minutes. So here's just another way of looking at the same thing. We're looking at our resting membrane potential, and here's kind of the axon membrane. And you can see that we've got all this positive ions or positive energy outside and negative ions on the inside, making this negative 70 millivolts difference between the outside and the inside. When we have a separation of charges like this, we say that the plasma membrane is polarized, meaning that the outside is more positive and the inside is more negative. And again, we're measuring this in millivolts. And you can see here that we've got these little channels, and these channels are going to allow the potassium, if this is the inside of that axon, these channels are going to allow the potassium to kind of leak out here. And we have some, and actually not as many for sodium, for the sodium to leak back in. But you're going to see a little bit later that we've got a lot more potassium channels that allows potassium to leak out. We also have what you've learned before, these sodium-potassium pumps. These sodium-potassium pumps, again, allow 3 sodium to come out of the cell and 2 potassiums to come into the cell. So between these two different types of pumps and channels, this resting membrane potential is going to be sitting at about negative 70 millivolts. And it's going to change a little bit. You might see some at negative 65, but somewhere between negative 65 and 70 is what we're going to call resting membrane potential. So I said earlier that that membrane is very permeable. You could think of this as being semi-permeable, but we're just going to say it's permeable right now. So we've got a high concentration of potassium inside the cell. So we got potassium really big here. If this is our cell, you can think of the potassium being inside the cell. You got a lot of it there. You got a lot of sodium outside the cell. And wherever there's a lot of sodium, you're going to find a lot of chloride that goes with it. That's our sodium chloride. So we've got both of those going on, but we said the cell is negative. So we got to have other things in here. And those other things are going to be anions. And those anions come mostly in the form of proteins. So remember, proteins with their R groups, a lot of them have a negative R group on them, and that's what's going to make this cell really negative. If you just had these anions in the cell's resting membrane potential would be negative 90 millivolts just by themselves. But because we added a lot more potassium in there and a little bit of sodium, that's going to bring that resting membrane potential up to negative 70 millivolts. So we said before that those ions are going to flow due to an electrochemical gradient. That electrochemical gradient then is made up of two things. We're going to have a concentration gradient, which is going to be our chemical gradient, and we're going to have it being those ions attracted to opposite charge. That's going to be our electrical gradient. So let's look at a cell and three different ions that we're going to concentrate on here. Chloride, we're going to look at sodium, and then we're going to look at potassium down here. So if we start with chloride ions, we can look at the chloride concentration gradient. Well, we got a lot of chloride ions outside of the cell compared to inside of the cell. So those chloride ions are going to want to flow down their concentration gradient to come into the cell. But there's also a lot of anions in the cell making this negative. So it's going to have an electrical gradient that's going to want to push them out of the cell. And it just so happens that if you were to think of the pressure of each of these gradients, they're going to be equal to each other for chloride. So here we've got 70 millivolts of pressure of chloride wanting to come down its concentration gradient, and we've also got 70 millivolts of electrostatic pressure pushing it out of the cell. So it's going to be staying in a net neutral here. When we look at potassium ions, they're almost just the opposite. One, they've got an opposite charge, so these are positively charged ions, and we can see that its concentration gradient much higher inside the cell than outside the cell. So those potassium ions are going to want to flow out to go down their concentration gradient. But this cell is negatively charged, so that's going to be pulling them back in. So if we look at those two pressures, we could say that we've got 90 millivolts of pressure of concentration gradient pushing them outside of the cell, and we've got about 70 millivolts of electrostatic pressure bringing it in because it's attracted to that negative ion. As a result, we're going to have a flow of potassium coming out of the cell because that concentration gradient overcomes the electrical gradient. So potassium's always going to be flooding out. The last one we have is sodium, and sodium, you can see, is kind of the opposite of potassium. Positively charged, high concentration outside of the cell, and low concentration inside of the cell. So both its concentration gradient is going to be pulling it into the cell, and it's going to be attracted to the negative anions inside the cell. So both of those are causing sodium to flood in, but they've got gated channels that are preventing that, and they have to wait until those gates open for the sodium to come flooding in. And that's what's going to be part of our action potential as we see that happening down on the axon. So you can see how we need a sodium-potassium pump to keep these guys in check. With the sodium-potassium pump, we're always putting that sodium out of the cell to keep that concentration gradient built up out there. And at the same time, it keeps the concentration gradient of potassium high inside of the cell. And we need those gradients to keep the cell charged at that negative 70 millivolts for its resting membrane potential. So we know that these cells can change their membrane potential very, very quickly, and especially neurons and muscle cells. They're going to really be able to change that rapidly. We call that being excitability or irritability of the neurons. And we know that it's being caused by that electrochemical gradient. So changing chemical gradient or the attraction to opposite charges. So we can kind of look at a graph like this and say, okay, here's our millivolts on the left side. We know that it's sitting at about negative 70 millivolts when it's at rest, but that thing is going to change. This is the excitability or irritability. And when it gets less negative, or you can think of it as becoming more positive, but it's not really positive till it cracks a zero, so it's less negative, we call that depolarization because it's getting less polarized. Remember, at negative 70, we had that separation of charges that made it polarized. Well, it's getting less so polarized. And it does that because positive ions are usually flooding into that cell. And usually those are going to be things like sodium. We call that excitatory. If you start then coming back down towards the resting membrane potential, we call that repolarization because we're getting polarized again, getting back down to that negative 70. And that usually has to do with potassium ions leaving the cell. So if you have positive ions leaving, it's going to make it more negative inside the cell. You could just as well have negative ions like chloride coming into the cell to make this thing less positive or more negative. And that gets you back down to resting. You can actually exceed that, and we call that hyperpolarization, because hyper means more, and we're getting more polarized, so it's really getting more negative. And so those, if you keep sending potassium out or keep bringing chloride in, when you reach that resting membrane potential, it gets hyperpolarized. And this is really inhibitory from creating that action potential. So you can kind of think of this here, if we just give you, I know a lot of you like different types of visuals. So if you think of this as being our cell, outside the cell and inside the cell, you can see we got a lot of sodium outside, a lot of potassium inside, negative resting membrane potential. Well, as that action potential goes, we're going to allow sodium to come flooding in here, and that's going to make the inside more positive. And then later we're going to have potassium leaving the cell. That's going to make the cell more negative. This is talking about action potential, but we could also be talking about these aren't action potentials. These are what we actually call graded potentials, which we'll talk about next, but they're just changes in this membrane potential. So you can see how potassium going out is going to make this cell more negative. Sodium coming in is going to make the cell more positive. That positive is called depolarization. The negativity when the potassium leaves is called repolarization. And then we use this sodium-potassium pump to bring us back into the balance that we need. A lot of sodium outside, a lot of potassium inside. That sets us up for getting our action potential, and we will start looking at the whole action potential in the next set of videos, so join me there.